So good afternoon. I think we can start. We're coming on to 2:15. So welcome to the Predicting Brain Age Across the Lifespan Models, Applications and Challenges Symposium. My name's Andrew Zaleski. I'm co-chairing this symposium with Dr. Tian from the University of Melbourne, as well as Tian. Has done all the heavy lifting in setting up the symposium, so I'd like to acknowledge and thank her for the hard work that she's put in setting up the symposium. We've got a fantastic lineup of speakers, both early career and imminent senior scientists in the field of biomedical image analysis. The core objectives of this workshop is really to understand the concepts of brain age and to convey to you the basic principles of estimating brain age using neuroimaging data, to understand the key methodological challenges and biases associated with estimating brain age from neuroimaging data, and also to give you some insight into applications of where brain age can be used both in neuropsychiatry and neurology. I'd like to remind you, if you have any questions, we'll have some time after each presentation. You can either come up to the microphones and ask your questions in person. Also remember that there's the app, and you can type your questions through the app, and I can read those out to the presenters after their presentation. So I hope you enjoy our symposium and that you get some understanding of the brain age concept. So first of all, I would like to invite Dr. Laura Han, who's from Origin at the University of Melbourne, to give our introductory lecture. Over to you, Laura.
Put this down a bit. Hi everyone. Thanks for joining our symposium. Thanks Andrew and for organizing this symposium. My name is Laura Han. I'm a postdoc researcher in the research group of Lianne Smaal, and today I have the honor of kicking off this session. And with that, maybe even if you might be familiar already with the concept of brain age, I'll start with a very basic explanation of some very basic modeling principles. So, to my knowledge, the first person to really pioneer and popularize the brain age concept is the neuroscientist Dr. Kawashima. And I remember back in 2015, I had this Nintendo DS and did brain training games, and you could do little speed calculations and memory puzzles, and then if your performance on these games would match those normally seen at younger ages, you could get a younger brain age based on your cognitive skills. And with modern advancements since, other scientists like Dr. James Cole and Dr. Katja Franke further evolved this concept by using supervised machine learning algorithms that predict age from brain scans. And I'll quickly run you through this process. Oh, do I have a pointer? Okay. Well, if you go to A, for example, if you take structural MRI scans from healthy individuals, we call this a training set, we will know the participant's age label, and we put those in a machine learning regression model that learns the brain patterns that are correlated to age. And then in B, to validate whether your model actually does a good job at predicting age, a proportion of the participant images are left out of the model. And for example, using tenfold cross-validation would involve training the model on 90% of your data and predicting age values on the left out 10%. And this is then iterated through all the participants, and the predicted values are then compared to the real values to assess the accuracy. And assuming that the model is sufficiently accurate and you're happy with the performance, the model is trained using the entire data set, and the resulting model coefficients are then applied to new participants' brain scans, or the test set, to generate brain age predictions. And so, for example, in C, based on their brain scan, this person is predicted to be 17.9 years old. And then the difference between a person's predicted brain age and their chronological age results in the brain age gap or the brain predicted age difference. And positive values may be interpreted as advanced brain aging, and negative values reflect delayed or healthy brain aging. And trained brain age models can be applied to any individual, but my specific population of interest was persons with a depression. And in this first part, I will talk about the application of these models to adult populations aged 18 to 75 years old. And for these studies, we have used data from the Enigma MDD consortium. So we had data available from almost 5,000 controls and about 3,200 patients. And instead of using raw T1 scans as input for these models, cohorts shared free surfer-derived summary measures based on the DK and ASEC atlases implemented in FreeSurfer. And we then averaged these across the left and right hemisphere and used these as input. We built separate models for males and females. And we split our data using 50% of the controls to train our models and the other 50% and 100% of the cases to test and validate our model and to get these brain age gap estimates and calculate case control differences. And so while higher dimensional features may lead to better performance accuracy, the model showed good performance when considering the mean absolute error were both about 6.5 years, scaled to the considered age range, and also good generalizability for the out-of-sample validation. Using this approach, patients with a depression were estimated to be about a year older than control subjects, but as you can see from these figures, the effects are rather subtle. And in other words, if given someone's brain age gap, we cannot reliably tell whether it's a control or a case. So this marker is not suitable to distinguish cases from controls. And so the estimated brain age gap in depression actually varies across studies in terms of effect size and statistical significance. And these differences are likely driven by difference in, for example, sample properties, so the age range, but also training or testing samples and the methods used, different features, different modalities. So when we had a subsequent addition of new cohorts to the Enigma MDD consortium, this provided us with a unique opportunity to perform an independent replication study in 13 new cohorts to validate our developed algorithm and also to determine the impact of depression on age-related brain patterns. And here we can see that the pooled metrics were comparable to those in the out-of-test samples in the original study. And using a mega-analytic approach, we were able to replicate the case control difference with a highly similar brain age gap and also associated effect size here of 0.15 compared to the previous 0.14 in the original study. In addition, we also performed a meta-analytic approach to kind of gain further insights into the generalization of the model in individual cohorts. And where I want to draw your attention to in this forest plot is that the small pooled effect size of 0.19 is not necessarily due to consistent small effects in all cohorts, but more of a result of moderate significant effects above 0.40 and the remaining non-significant effects. Important to note here is that we were able to examine some basic clinical characteristics pertaining to, for example, antidepressant use or recurrent episode status, but found that these were not able to explain the variation in the brain age gap. However, in terms of scan technical moderators, we found that, for example, image acquisition with a Philips scanner showed significant moderating effects on the effect size of the case control difference in the brain age gap. While a scanner manufacturer may potentially lead to differences in morphological estimations, I think it's very unlikely that effects will be opposite in cases and controls. So it seems more plausible that other heterogeneous demographic or psychosocial or biological cohort-specific characteristics coincided with this scanner vendor variable. So from these studies in adult depression, we may conclude that two large-scale, independent but harmonized mega-analyses across 32 cohorts and about 3,200 patients and 2,700 controls show a reliable but very subtle pattern of brain aging in adult depression. However, the small pooled effect size was not due to consistent small effects across cohorts, but may be in part driven by the heterogeneity across scanning sites. Using the limited harmonized measures that we had available, we did not find a relationship between basic patient properties and effect size differences, but obviously more work is needed to examine which characteristics, clinical and biological, may underlie the individual variation. And this is exactly what we and other researchers in the Enigma Consortium are currently working on. So you can expect more work to examine what kind of clinical and biological factors contribute to the underlying variation. So for part 2 of my presentation, we shift our focus to adolescence and young adulthood, a period of peak onset of mood and anxiety disorders. And adolescence is a period where the brain is still developing, but while in young adulthood, we may already see some signs of aging. And so if we are attempting to truthfully capture age-related brain changes, ideally separating these two concepts, the classic whole brain age paradigm might be suboptimal. However, the potential clinical relevance is understudied, and examining whether the brain age gaps in adolescents are associated with mental health and predict disease severity and treatment response is not necessarily dependent on this first attempt, and worth testing in my opinion, even if it simplifies or doesn't truthfully capture brain age trajectories or brain trajectories. And so we gathered 3 cohorts in Melbourne, totaling to about 250 controls and about 240 patients with either depression or first episode mania. And to calculate the brain age gap in adolescence and young adulthood, we applied a model developed by Nusha Diastani, who also works in our lab. And this model includes about 1,100 subjects from the HCP development and young adulthood cohort, and also relies on free surfer ROIs, but also includes left and right hemispheric measures separately, as well as some additional cortical and subcortical volume measures or features. And she used the support vector regression with a RBF kernel, and under tenfold cross-validation found a mean absolute error of 0.28 years, or translating to about 3.3 months. And as you can see here, Nusha's model showed good generalization to these independent clinical cohorts. But when comparing the brain age gap between cases and controls, we found no significant differences. And also, when we looked at depression severity scores in the patient groups, we did not find a significant association with symptom severity. And another objective was to assess whether the brain age gap could predict treatment response, which was defined here as a 50% reduction in symptoms. But as you can see from these preliminary analyses, the distributions of both groups overlapped, and a logistic regression showed that the brain age gap could not predict responders from non-responders. It's good to mention that we're currently harmonizing more clinical measures across the 3 data sets, so more work in this area is also forthcoming. Some preliminary conclusions. So we trained a brain age model specifically tailored to adolescents and young adults, and despite the good model generalization, we did not find associations with depressive psychopathology or treatment response. And given the subtle effects that we saw previously in the adult populations, this absence of the case control effect is perhaps not very surprising. It might be that the age-related differences in adolescent depression are quite or rather limited, but it could also be that the aging process is not necessarily the main process that has an effect on brain structure in this age range, and we might need to consider other factors, such as puberty, for example. But these preliminary findings do seem to beg the question at least whether a whole brain single metric of brain age is sufficiently sensitive. So our next steps are to train higher dimensional models and combine different features and modalities, and also given the region-specific developments of the brain, we will also model local brain age algorithms, and also have a look at longitudinal models that are more, would maybe shed more light onto within-person brain age changes. So if you're interested, or if you're also working on adolescent brain age models, I'd love to have more discussions with you on this. Yeah, and also by the other speakers will definitely touch more upon this, I think. To end my presentation, I recently got gifted a Nintendo Switch with a new version of the brain training game. And without disclosing my actual age, I'm pretty proud to share that I was able to modify my plus 28 years now to -8. So if anyone in the room is willing to put me in an MRI scanner, I'd be very keen to find out if my brain age is the same according to the presented algorithms today. Yeah, thanks everyone for listening, and of course many thanks again for organizing. And I want to thank all my collaborators, because of course I did not do this work alone. And I want to thank all the Enigma MDD contributors and everyone that has supported this work. And I'm happy to take any questions if you have them.
Thanks very much, Laura. That was a fantastic overview of some of the clinical studies using brain age, the brain age concept. So we have some time for questions. Perhaps we can take one question from the audience, and then I can read out one of the online questions. Just a reminder that you can post questions online and also upvote questions that have been posted. Also, for those that have come in late, there are still plenty of seats in the front row. So if you want to come to the front, you're welcome to come up and take one of the seats. So are there any questions in the audience for Laura? So if not, perhaps I can read out one of the questions that have got upvoted. One of those is, how stable or reversible are the brain age gaps in clinical groups? And I think this is in the context of recovery. So can we reduce the brain age gap?
Yeah, of course, that's the hope and promise, that it's actually a modifiable metric that can be influenced by interventions, for example. I think until now there isn't much work or much evidence that seems to suggest that this is possible. Looking at different biological age indicators that are modeled more or less the same way, but using different biological data, there is some preliminary evidence that this could be possible, at least maybe not reversible, but at least been brought to a halt. I think Fidel will talk a little bit more about this as well, whether it's a thing that is set very young in age and then doesn't track the range of brain aging per se, but I think he will answer to these questions much more than I can.
Thanks. I think we've also got a question here from Martin.
Hi, thanks for a fantastic talk, and I think we're all jealous of your -8. Do you know whether there are also other signs or other aspects of the body that this brain age relates to? Were these also healthier people? Were they more fit? Was their skin younger? Can we have a really young brain in an old body and vice versa?
Yeah, so in the work that I've also been involved in, looking at different, like an integrative study looking at different brain ages in the periphery, so based on different omics levels, for example, we actually found that most of these biological clocks are unrelated to each other, or at least very weakly, if they are correlated, very weakly correlated. So indeed, this seems to kind of suggest that you could be younger in one biological system, whereas you could be older in the other one. I think one of the advantages of these things that they're not very well correlated would mean that if you combine these measures together, they actually explain a different part of your biological system, and you could potentially be older at multiple biological systems and then potentially have more cumulative aging, for example. And this was indeed also had stronger effects, so yeah.
One more question.
Can I do another one? Or
maybe one more question, a quick one that's been upvoted a lot is why did you use two separate models for gender?
Sorry?
Why were there two separate models for gender, one for males and one for females?
Yeah, we actually followed the Kaufman paper, and also with the idea that males and females potentially have different rates of development and aging. Therefore we had a lot of data to actually build separate models for these, and that's why we took that rationale.
Makes sense. So please join me in thanking Laura Hahn once again. So our next speaker needs no introduction. Professor Christos Devadzikos is an international leader in biomedical imaging analysis. He's made many seminal contributions in developing and applying machine learning approaches to establishing biomarkers for both neurological and neuropsychiatric disorders. So it's a real pleasure to have Professor Devadzikos join us today, and I'm really looking forward to his presentation. So over to you, Christos.
Thank you so much. It's a great pleasure to be here, and thank you for organizing this great session. So you heard about brain age. I'm not going to reiterate that. You fit a model, a multivariate model, and then you take the residuals, and obviously it's a very useful measure, as you saw from Laura and in all the studies that has been shown to show significant effects in a variety of areas: schizophrenia, AD, depression, sleep disorders, MCI, social anxiety, a number of other areas. So it's certainly a very useful measure. I'll talk a little bit about the caveats in how we build and particularly evaluate some of these models. And there's been, because it's a useful measure, brain age has set off a race for, my model is the best kind of, especially in the technical world, and where people measure mean absolute error, and they say, oh, my error is 1.5 rather than 1.7, so my model is better than yours, and they use even higher multivariate models to achieve even better MAE, and I wonder if that's what we should be after. if we're looking at brain age as a biomarker of pathology, which is what we really want to. So tightly fitting brain age models might not produce the best disease biomarkers. And the question is, how well should we fit the models to get meaningful biomarkers? So I apologize for stating the obvious, but I like to go through a couple of cartoon slides that demonstrate the principle. So this is, say that the brain age model picks up these gray areas as are being informative for estimating age. But it happens that there's an asymptomatic pathology A that affects that part of the brain. We all have stuff in our brains as we get older, so, like myself. We're almost certain to have a number of pathologies in our brains. It has been shown that the prevalence of stroke is 5 to 10 times higher than what we see clinically. So as we age, we all have small vessel ischemic disease, we have Lacunar infarcts, we have, Lewy bodies, amyloid, all sorts of things that are not really, you can be cognitively normal and, typical aging person, but you have a bunch of stuff in your brain. Your brain age algorithm is going to try to avoid this area because that reduces that increases the MAE. It doesn't give good predictions. Another pathology can affect this part of the brain and another pathology that part of the brain. So the algorithm will end up focusing, if you really try to get the best fit you can possibly get to win the challenge, you will focus on things that are potentially irrelevant to all of these pathologies. Some of the answers to that has been, or some of the claims have been that we should be using very normal cohorts, super normal cohorts for building brain age models and then apply them to disease populations. But number one, it's very hard to find very normal cohorts. Basically, it's almost impossible to find, particularly in aging cohorts, to find people who are completely normal and to know that they're completely normal. As I said, we all have, as we age, we all accumulate stuff in our brain that we don't want to have, but we do. But that stuff doesn't necessarily manifest itself as through clinical measures. So, and then even if we were able to find a super normal training set, if you want, would that be informative of the pathologies we're interested in? Because in the end, we want to apply brain age models to a number of non-normal clinical populations. So. We played with these ideas a little bit a couple of years ago. This is one of my students, Vishnu Bashyam, a bioengineering student, who we had a paper in Brain 2 years ago where at the time, he used one of the sort of state-of-the-art deep learning networks and Inception ResNet. He adapted it a little bit, but he didn't do anything particular to fit he didn't optimize the network, he didn't really pre-process the data, he didn't skull strip the data, nothing. He just basically applied it to one of the consortia we have, the iStaging consortium, where we have put together, in this case, it was about 13 different studies shown in different colors on the left. And these are cross-validated brain age estimates, so we're pretty happy that it does fairly well. And on the right, you see an independent test from the SHIP study in Germany that the predictions of the model were pretty good. So we're pretty happy, and we started asking the question, what do the brain age residuals tell us in when they are applied to populations with AD, with MCI, with schizophrenia, with depression, and so forth? And what we found is what we hypothesized, that unfortunately I don't have a pointer, but the top rows here show effect sizes and histograms of brain age gaps for loosely fitting models. The middle rows are for moderately fitting models, and the bottom rows for tightly fitting models. And what you see consistently across everything is that the middle rows produce stronger effect sizes and higher significance than either the top or the bottom row. So in other words, if you overfit the data or if you underfit the data, when you apply the model to clinical populations, you get less power to detect disease effects. So the message here was very clear from everything we studied, and this is only a few of them, that you shouldn't overfit or underfit your brain age models. You have moderately fitting brain age models. So the bottom line of this is that if you're interested in estimating age, look at the passport or the birth certificate. If you're interested in a disease biomarker, do not really evaluate MAE, but look at the brain age residuals in the context of what you're trying to do, whether that's diagnose, predict response to treatment, and so forth. It's still painful to see bunch of challenges, especially in the Mackay world and other areas where people are trying to beat the best MAE out there. So another issue that we have to have in mind, again, I'm talking about caveats. That doesn't mean that brain age is not a good biomarker. We have to have in mind certain things as we use it as one of many possible in silico biomarkers if you want. Another thing we have to have in mind is that, say that we have a brain age model that uses the gray areas to estimate age, and we take the residuals, and then now we apply it to a pathologic population which has pathology in this part of the brain. It's going to trigger the brain age models. It's going to produce presumably higher residuals. However, it so happens that another disease affects the brain in a way that doesn't happen to overlap with this model. So this is not going to be detected. So we might conclude that, oh, this population is normal. But it's only normal from the perspective, from the projection, if you want, onto the brain age model. It's not normal if you were able to measure everything in the brain. So it's an issue of sensitivity versus specificity, but again, I'm restating the obvious, but oftentimes we tend to forget that if we have a negative finding, it doesn't mean, in this particular case, that the brain is normal. It means that the brain age residual was not able to detect it. If we do detect something, then we're more certain that something is there. On the more positive side now, what we found out is that brain age models are really very good at pre-training, particularly deep learning networks. Those of you who work in deep learning know that you have to pre-train your network before you apply it to a particular classification task. Say you're trying to classify AD or MCI or schizophrenia or anything else. And you have, a set of 500, which is not sufficient for training the model. You have to pre-training, and people typically use something like ImageNet, which has tens of million or over 10 million images to pre-train the model, and then do the final training on, say, schizophrenia or AD. What we found is that if you were to pre-train your network on brain age, where just because age is widely available for tens of thousands of MRI scans, so you can pre-train your network on brain age and apply it to these smaller end clinical classification problems, you do consistently better, as the blue lines show here compared to the green. So green is training on ImageNet, blue is training on what we did. We trained on over 10,000 brain MRIs for age, and we found that consistently when you apply it to clinical populations, especially if you have small n, you do better. So certainly, and this is a model we made available after this paper to the community if anybody wants to use it. We now have much larger populations, so it's a model that one can take as pre-trained model and adapt it to their own population if you want, or clinical problem. Another issue that we have to have in mind is heterogeneity. Again, I have highlighted 2 points here in red, that these are people who have high brain age residuals. However, they might deviate from the norm for different regions. They might be very different. So, it's an issue of specificity of brain age. It might trigger abnormality, but it doesn't tell us what type of abnormality you have. This is a simple example of some work that's been going on in my lab recently, trying to demonstrate that by Divya Srinivasan, who is one of our research staff members, and Matilda Antoniadis, who is a postdoc, a senior postdoc. So they just try to take a very simple problem, structural and functional brain age. Say that you're only looking at, and then they basically derive the two-dimensional system where I cannot point, but the y-axis is structural brain age, the x-axis is functional brain age, and say that you're only looking at structural brain age, and you don't really have resting state fMRI, you would not be able to differentiate between the orange and the red, just because both of them have high brain age. Some of them have, structural brain age. Some of them have, though, advanced functional brain age, and some of them have resilient functional brain age, and they have big differences. If you look at the orange and the red on the right, they have very different rates of cognitive decline, even though they have similar structural brain age. If you look at other measures here, they're also somewhat different. Now, conversely, if you only have a functional brain age measure and you take the outliers, you cannot differentiate between the red and the green. because they both have high functional brain age residuals, but you haven't measured the structural brain age residuals. Well, if you look at these people, they're quite different. For example, the red and the green at the top left, the green has significantly higher accumulation of amyloid at earlier ages than the significantly lower amyloid accumulation than the red folks. The green and the red have different rates of cognitive decline, and they have other differences, particularly the SPARE-AD an index we've developed over the years for Alzheimer's that shows that the people in green, so the people who, even though they have advanced structural brain age, they have resilient functional brain age, they also have low SPARE-AD scores, so they don't present AD-like patterns of brain atrophy. So again, these are only two axes, but if you have missed one of them and you only look at structural residuals or functional residuals, you'd really miss the picture of the other one. So projections are only good for certain things. So for this reason, we've been spending quite a bit of time in my lab on the methodology side to develop the smile gun method, which we published in Nature Communications recently. This is Zizang Yang, one of my math graduate students, and we showed that if you break down the heterogeneity of brain aging into, or particularly in MCI and AD individuals, into multiple dimensions, multiple patterns, you can predict the progression to Alzheimer's disease quite well, as you see on the right, with 95% confidence intervals. So again, you need multiple dimensions to really pick this heterogeneity. So we've been doing that in Ioanna Scambardoni and others from my group have a poster here. We're doing that to try to pick up, to dissect the heterogeneity of brain aging and kind of see people who have high brain age gaps, but in what different ways do they deviate from typical brain aging? Because one number doesn't say it all. So the final point I want to make is size and diversity. We really need to train on a lot of data, and particularly diverse data. And people like to, I hear big data more than I hear diverse data. And I emphasize diverse data is more important than big data. And diverse in many ways, both in terms of the composition of the cohort for race and sex and other covariates, but also in terms of scanner types and protocols and so forth. And towards this end, I'm spending a good part of my time over the past 5 years, and I see Mohammed Habis here and Ahmed Abdul Qadir. They are senior people in my group, or formerly in my group, who have spearheaded some of these consortia, trying to put together data from 17 different studies, 65,000 individuals, and try to train brain age and other models from much larger populations and diverse. And this is a consortium we recently got funded with a number of us. You see some of the faces here who have been leading this consortium, trying to bring together data from 50,000 individuals from around the US with imaging, genomic, and phenotypic data to be able to learn and train these models on much more diverse data. So to summarize, we need to evaluate brain age models in the context of the task at hand and not in terms of MAE or some other quantitative formulaic measure. Whether that's diagnosis, prognosis, response to treatment, whatever you want to do, your brain age model that gives the best biomarker might be different. Brain age models can serve as very good pre-training models, especially for deep learning, but also for auxiliary learning. I didn't show you some of these results, but we have a lot of fun with auxiliary learning, meaning that you have a main task, say schizophrenia or Alzheimer's or depression, and you have an auxiliary task, age, that helps you fine-tune your models and your features and everything else so that you can both extract the right features and adapt to new domains. So they're very good for that because age is widely available for tens of thousands or hundreds of thousands of people with MRIs. So we can really use it to adapt, whereas clinical status might be less available, or genomic data. And finally, a single measure, the most important message is that a single measure doesn't say it all. Heterogeneity requires richer and more diverse set of biomarkers. Brain age measures are one set of them, but I think in the end we're going to be thinking about, and I think there's other symposia tomorrow for brain charts. We're spending a lot of time on, trying to tease out multiple dimensions, not just one or two, multiple dimensions from using these machine learning models that will eventually help us condense complex data down to maybe 12 or 20 or 30 dimensions, not one or two, but not millions either, so that we can actually evaluate disease effects, trajectories in these brain charts, and kind of find disease subtypes or predict response and so forth. So thank you very much, and I'm happy to take any questions.
Thank you so much, Christos, for the fantastic presentation. We've got so many questions here, so I'll only read out a couple of them. The first one is an interesting question. It's a very simple question. What does it actually mean to have an older or a younger brain? So at the end of the day, what we're looking at here is residuals or error terms in a model. So how can we then extrapolate to make the conclusion or make the inference that these relate to age or aging?
Yeah, that's exactly one of the points I try to make, that it doesn't necessarily mean, it's not very specific of what exactly you have. It depends on how much your pathology intercepts the model. If it doesn't happen to intercept it, you haven't picked up signal. So it doesn't mean much, but it can trigger further evaluation if you want. It's also a convenient, simple or single measure of brain health, which kind of summarizes things. But it certainly by itself is not sufficiently specific.
Are there any questions in the audience? Perhaps we can take one question for the audience. Otherwise, we can read one out.
Please just use the microphone when speak out.
Thanks for the talk. Did you ever look at disease subtypes and how maybe some, for example, for Alzheimer's disease, correspond with this change in brain age, and maybe some don't fit that model as well?
Yeah, I don't know if I can go back. It doesn't matter. I showed you, I went over one of the slides. We found in MCI and AD there are 4 subtypes of brain atrophy patterns using this SMILE GAN approach. And they don't trigger the brain age gap the same way. But one of them has a relatively modest atrophy that would really give fairly normal brain age residuals, but it's the one that is very focal on the hippocampus and medial temporal lobe, and that's the one that has one of the worst prognosis. So it's a perfect example of that you can have a pattern of brain atrophy that doesn't necessarily trigger your brain age residuals, but it is highly indicative of some pathology like Alzheimer's.
Thank you.
Sure.
So perhaps one more question from the online app is, what's the benefit of brain age prediction models for clinical use over pathological feature models for prediction? So what are we gaining by using brain age as opposed to using raw phenotypes?
Yeah, I mean, I think I see brain age more as a screening tool. If something comes up, you need to look at it more carefully. So if your brain age is higher, that means that there's something happening in your brain somewhere. You cannot tell exactly, but you should, do either more analysis, or you should do on the MRI or more clinical phenotyping and so forth.
Well, thank you very much, Professor Christos Stavadzikos, for the
Thank
fantastic
you.
presentation. Please join me in thanking Christos again.
Thank you.
So our next presenter is Dr. Vidal Pinheiro, Dr. Didac Vidal Pinheiro from the University of Oslo. Didac has done some fantastic work on characterizing differences in aging, or differentiating the effects from aging from early life and developmental factors. So looking forward to your presentation, Didac.
Thanks. First, I would like to thank the OHBM organization and Tian and Andrew Stalensky for putting up this symposium and having the opportunity to present our work, in which we try to understand how better to interpret variations of delta in cognitively healthy populations. So brain age residuals are a quite common and generic measure that is often understood as a biomarker of individuals' brain health. And in the last few years, aging research has repeatedly shown how higher delta has been associated with worse outcomes in both cognitive function, clinical, or health in aging populations, including, for example, survival probability. This and the fact that it reflects variation in structures that are quite sensitive to age, it leads to the common assumption that it must reflect ongoing neurobiological processes. That is, that delta, it is a measure that is capturing or it's sensitive to the rate of brain aging. However, this is not necessarily the case, as delta, it's in one way or another, it ends up being residualized. So it reflects deviation from the expected norm akin to normative modelings. It can then also reflect like pre-existing differences in brain structure that have earlier origins. So in this simplified scenario, we can interpret findings of delta with a given outcome in two possible ways. For example, we can say that it reflects the slope differences in the rate of brain aging, shown here by these differing lines that mean that two groups have different trajectories or ongoing differential brain change. This means that at older age, variations in delta will reflect, will be influenced by the rate of brain aging, and it will be also, like, it will predict as well future ongoing change. Else, it can be reflect intercept differences and differences in delta can have lifelong stability, meaning that these differences can emerge earlier in life, and then we have quite similar lines or slopes of brain decline. When you go to older populations, would show a similar effect for a given outcome, and if you look at future sensor-based outcomes such as dementia, you would not find a difference. With cross-sectional data only, it's very difficult to disambiguate with these two possible interpretations. So with this in mind, we aim to use longitudinal data to test these two possible interpretations. And for the first one, if delta reflects the rate of brain aging, we argue that we would find a relationship between cross-sectional delta and longitudinal brain change. Here in the figure, you would see a relationship between the distribution of the points, meaning cross-sectional data, and the slopes of change, the arrows. We also argue that delta values should spread with higher age, meaning or being an indirect measure that individual delta changes with age. Rather, if delta reflects lifelong stability and has early life influences, we assume that variations in delta, cross-sectional variations in delta, would be related to early life factors known to have, that are supposed to have a quite stable influence on brain structure throughout the lifespan. Here we use normal variations in birth weight and polygenic scores for brain age. So to do this, we use the UK Biobank as the main data set and the live brain as a replication sample. We use, for the training data set, we included all participants that had only a single observation, while the test data set was, we used for the test data set individuals that had at least 2 or more MRI observations, so they had longitudinal MRI data. You can see the age distributions on the right side of the image, and we end up with 1,400 and 1,500 unique individuals for both test data sets. Live brain data set was a little bit, had a longer follow-up and more observations on average, but also a wider age range. So we estimated quite standard brain models on the cross-sectional only data. For the main analysis, we use a gradient boosting algorithm and a lasso algorithm as a technical replicate. And we use a randomized hyperparameter search and tenfold cross-validation on T1 weighted features showing regional and global features of cortical thickness, volume, area, intensity, and gray-white matter contrast. So the models were then applied to the test data sets, the longitudinal data sets, and show more or less comparable to performance to what has been reported previously using traditional machine learning algorithms. To To test the relationship between cross-sectional delta and longitudinal brain change, we use the centercept for the cross-sectional delta and 3 measures of brain change. The first was longitudinal change in delta, and the second was first main component of longitudinal change in all features using a principal component analysis. And then finally, we use as well change in all the features that were introduced in the brain age estimation. So first, did we find the relationship between cross-sectional and longitudinal delta? And the answer was no, we did not find any kind of relationship, neither in the main test, neither in the 2 replicates. Actually, we found a negative relationship between higher delta and longitudinal change in delta over time in the main dataset. Then we use equivalent test to try to reject effects of a size x, and results show that essentially if there exists a relationship between cross-sectional and longitudinal delta, it explains at most, or we could reject variances explained over 0.2% of the variability. Next, we replace the longitudinal measure by this main component of change, and likewise, we did not find any significant relationship between cross-sectional delta and longitudinal brain change. And when we use all the single features that were inputted in the brain age model estimation, here, yes, we find indeed a relationship between cross-sectional delta and a ventricular expansion, which was replicated in all the different models. Finally, we also test whether delta variations in delta spread throughout the dataset's age range using Smith formulation in the Neuroimage 2019 paper. The results show that delta does spread a little bit across the age, but just quite modestly. Overall, it spreads around 0.4 years throughout the dataset's age range. So since 18 to 18 the live brain, and from 45 to 80 something in the UKB biobank. To sum up this analysis, we show that they show that delta variations in delta, cross-sectional delta, do relate to ongoing rate of brain aging, but just quite modestly. This is quite related as well to, it is related to earlier findings where it has been shown that there's a poor correlation between cross-sectional estimates of MRI and longitudinal changes. Then next we test the second possible interpretation, and whether delta variations in delta were associated with early life factors. First, we looked whether it was associated with self-reported birth weight, and we found in it that cross-sectional delta was correlated with normal variations in birth weight, both in the main dataset and in both replications. The relationship was quite stable on how we define normality of birth weight. And then we also test whether variations of delta were associated with polygenic scores of brain age. We computed polygenic scores based on a GWAS analysis on the training dataset, on brain age on the training dataset. We found a cross-sectional relationship that cross-sectional delta was associated with genetic liability to brain age, but we did not find a relationship between change in delta and genetic liability for having a brain age. In both analyses, if we include intracranial volume as a covariate, we found quite significant but quite small effects. But we think this is also kind of a proof of concept on where we can find quite meaningful variations of brain age in older populations. So just to conclude, delta variations, cross-sectional variations in delta do reflect to a very modest degree actual rate of brain change in middle to adulthood, but most of the variance actually is probably present before the age's lower bones, and some of them must reflect early, or seems to reflect early life influences on brain structure. these results most likely are quite dependent on specific samples, features, modalities, algorithms. But yeah, we would like to end up with a note of caution when interpreting brain-derived indices of aging based on cross-sectional MRI data as indices of ongoing and future brain change. So most of this analysis has been published in this paper, and you can check them out as well in this poster that is hung today and tomorrow in the poster hall. I would like to thank my collaborations in the University of Oslo and elsewhere in the world, also the funding agencies and Biobank and LifeBrain for providing the data. Thank you very much.
Thank you very much, Didac, for the presentation. One of the questions that's come up in several forms is how would these results look if you were to train the brain age for a particular region, or even taking it to an extreme for a particular voxel? Would you still see this distinction between early life versus the aging concept? So, for example, here we've got only one measure for the whole brain, but can we do this, for example, for the hippocampus or even for a particular voxel?
So, yeah, probably. So you mean like feature versus vertex-wise?
So rather than using the whole brain as our feature space, our feature space would be a specific region or even a specific voxel.
I mean, of course, the specific results might change, but I don't believe the conclusions of these results would change a lot. I would expect more variation if you change, for example, samples, or if so. These results might be slightly different if you change, for example, the sample in which you train the dataset or the sample in which you are applying the dataset. But still, you know, if you apply it to Alzheimer's disease patients, most likely, and you would find a relationship between cross-sectional delta and ongoing change. But I don't think that the cross-sectional data per se allows you to answer this question. I don't think that vertex data or vertex-wise data would improve a lot.
Makes sense. So maybe we can take one quick question for the audience, from the audience, if there are any, before we move on to the... Yep.
Hi, we are also looking at the UK Biobank, and it seemed like the people who came back for the second imaging were possibly younger to begin with or healthier to begin with. So then I was wondering if there could be this kind of bias for this longitudinal data collection, and if this could affect your analysis. Thank you.
Thanks. Yes, I don't think these results should be quite affected by survival bias or biases due to dropout in follow-up, because, but of course there could be like a small influence, but I think that at the end of the day you are comparing like the structures of covariance between inter-individual variation and within individuals, and I would guess the effect is quite small. In the UKB dataset, for example, in terms of age range, they are quite similar. I think maybe the effect might be slightly, if any, would be a little bit more on the LifeBrain dataset. But yeah, I'm doubtful. But, yeah.
Thank you very much, Didac. Please join me in thanking Didac once again for the fantastic presentation. So our next speaker is Dr. Tian, who is also a co-organizer of the symposium. In fact, has done all of the hard yards in setting up this symposium. So all the credit to Ye's from the University of Melbourne, and she's going to present some of her new work where she's extended the brain age concept to other organ and body systems. So over to you, Ye.
Thanks, Andrew. Thanks for the nice introduction. So, and also thanks everyone for coming to our symposium. I think after the previous 3 lectures, most of you should have had a fairly good understanding of the principles of brain age prediction and know how to properly interpret the brain age gap indices. So in the last lecture of our symposium, I will talk about the application of brain age prediction models in the broader field of biological aging. So that's why I call that crosstalk between brain age and the body age. So this figure just shows that, as people might know, that the biological aging of human brain has been the focus of considerable research in the last decade, as indicated by the almost exponentially increased number of publications in the last 10 years. So indeed, so the brain age index has becoming a quite useful biomarkers to facilitate our understanding of brain aging and the brain disorders. However, the brain is not the organ system that is strongly affected by the aging processes. As stated in the stereotype hypothesis, aging is the result of deterioration across multiple organ systems, and that this deterioration is the root cause of age-related diseases. So indeed, the multi-system morbidity is pretty common in older people. Just a few examples. For example, people with schizophrenia, when they are getting older, they have approximately twofold increased risk of developing cardiovascular disease and diabetes than the general population, whereas the cardiovascular disease is also a strong risk factor for dementia. So if we can look at this picture, so given that the organ systems are interconnected by circulatory, nervous, and lymphatic networks, so we think it is likely that brain age would affect the aging of other organ systems, and similarly, the other organ system aging would affect brain aging as well. So to understand how brain and body interact as people age, in this study, we adopted the brain age prediction framework to other organ systems. We also did that for the brain and other 7 organ systems, including the immune system, cardiovascular system, hepatic, pulmonary, musculoskeletal system, renal system, and metabolic systems. So in the study, we used a subsample of UK Biobank participants that was completed a physiological assessment for body function across multiple systems, and also individuals with completed multimodal brain imaging, including the T1 weighted, diffusion weighted, and functional MRI. So we started with, according to the very comprehensive medical information available from the UK Biobank, we first define a group of healthy individuals, and also, as mentioned in previous talks, we train the age prediction models using organ-specific phenotypes to predict the age, such that once the model is trained, we use the trained model to predict the age of individuals with chronic diseases, such that the differences between predicted and chronologic age, often called age gap index, will quantify the extent to which a person's organ system appear older or younger than the population norm. So in this study, we use organ-specific phenotypes, and then we estimated the biologic age for each organ system and for each person. And then we mapped the biologic aging profile across multiple systems in 16 chronic common brain and body disorders, and we investigated the impact of genetic environmental factors on the aging. And then we also investigated the mortality risk associated with biological organ aging. Okay, so this picture again, one of my favorite. As you see, that given that the body and the organ system are interconnected by the circulatory, nervous, and lymphatic networks, one of our hypotheses is that an organ's biologic age would influence the aging of other connected organ systems. So to test this hypothesis, we first used the structural equation modeling to model the directional influence from the baseline body age to the follow-up brain age. As you can see on the left bottom, because of the time for body and the brain assessment were not at the same, such that we can have the time flow from baseline body age to the baseline brain age, but actually it's a few years later. So given this time flow, so we use the structural equation modeling to model the directional influence from baseline body age to the follow-up brain age. So this picture, this graph is a bit complicated. So I just want to highlight one of the findings. So here it basically shows that the cardiovascular age demonstrates the strongest influence on brain age, where a 1-year increase in cardiovascular age explains approximately 27-day increase in overall brain age, and also 19 and 27-day increases in functional connectivity and white matter age, respectively. So we next then still use the structural equation modeling, but we take advantage of the baseline and the follow-up assessment for both body and the brain function. We model something that already mentioned in Didak's work we use the structural equation modeling to model the influence from the baseline organ age to the longitudinal rate of change in the organ age of other organ systems. So this then gave us a multi-organ aging network. So I just to highlight several examples. For example, so in this network, what we show is that advanced pulmonary age leads to faster cardiovascular aging, which in turn leads to faster renal and musculoskeletal aging as well. As for people, people from, as we are doing neuroimaging, so from the network science perspective, as we can say that the musculoskeletal system is an integrated hub in this aging network, so it suggests that a faster musculoskeletal aging might be a shared consequence of aging across multiple organ systems. Similarly, for the brain, we say that advanced gray matter age leads to fast aging of the brain function, but not the other way around. We also see a positive feedback loop between functional connectivity and white matter aging as well. So as you can see that we cannot, because of the brain and the body function were not assessed at the same time, so we could not model the same network between body age and the longitudinal change in the brain age. But this hopefully can be addressed in the future when data is available. So here I just listed the 16 chronic brain and body diseases were studied in this work. So they are either have lifetime contribution to brain-associated illness burdens, such as depression, bipolar disorder, and schizophrenia, or they contribute significantly to health burden in older adults. And here, this is a picture. That figure just shows the distribution of the estimated organ age in each disease group. The first row is the comparison to the healthy group. So all the distributions, so now they are colored according to the mean age gaps to the disease-specific and organ-specific age gap in each group. So those distributions colored in gray, they just means that they're not significantly different from the healthy people. So what we generally find is that, so for the organ systems that are primarily implicated in disease pathology generally appear the oldest. For example, we say that the pulmonary system aged most in chronic obstructive pulmonary disease. The renal system aged most in chronic kidney disease. The hepatic system aged most in cirrhosis. And the metabolic system aged most in diabetes. However, this is not the whole story. Even though for many common diseases where the organ system not typically implicated in the disease processes, they also show significant advanced aging. For example, here I just show you a very simplified clock-style version to only show the mean of the age gap in each disease group. So as you can see that although while the brain aged the most, in the most common major brain disorders, including the musculoskeletal disease, dementia, Parkinsonism, individuals with non-brain disorders, they also show significantly advanced brain age, such as diabetes, chronic kidney disease, COPD, and cirrhosis. So similarly, for the body system, we see that while the common, very chronic, devastating body disease, including the chronic kidney disease, cirrhosis, COPD, diabetes, their body system aged the most. Individuals with brain disorders, they also show significantly advanced body age. Their body also appears significantly older than the general population. So just as a brief summary, in this work we show that the chronic diseases are characterized by unique organ aging profiles, and also across the 16 common chronic diseases we studied, advanced biologic aging extends from the organ or primary disease pathology to multiple organ systems. In terms of the mortality risk associated with biologic aging, we started with a very simple analysis, just a group comparison of the organ age across different systems in those deceased individuals and non-deceased individuals. So we used the mortality data ascertained on the 4th of March 2021. As you can see that this is quite consistent that people, like deceased individuals, even at the baseline assessment, their brain and the body already appear older than those non-deceased individuals. We next use Cox regression. We then find that even though after controlling for many factors, including the chronological age, sex, and existing disease burdens, and also those common lifestyle risk factors such as smoking and deprivation, we still see significantly increased mortality risk associated with advanced body age. For example, the bar plot shown on the left, what it means is that if we look at the second, look at the third bar, which indicates, which shows that each one standard deviation increase in the pulmonary age associated with approximately 10% relative increase in the mortality risk. And we see a significant increased risk, mortality risk associated with the overall body age, the pulmonary age, renal, and hepatic age. However, this is not the case for the brain age. In our study, we did not find significantly increased mortality risk with advanced brain age. While this is contradictory to a recent report in 2018 by James Cole and his colleague, we think one of the explanations might be because of the much lower mortality rate in the UK biobank participants with brain age estimates, because the brain imaging were only acquired very recently. So in order to have better understanding, I think the continuous follow-up will be very important to give us a better picture whether the brain age explains significant mortality risk. And finally, as I mentioned, because we see the increased risk associated with body age, so in this analysis, we trained a predictive model using logistic regression and tenfold cross-validation to accurately predict a person's survival time and premature death after the assessment of their body function. So what we find is that, so model 3, we find that even though the chronological age, sex, and existing disease burden can already explain some prediction accuracy, further incorporating the body age estimates improve the prediction accuracy further. So which means that the body age has some unique contribution to the mortality risk prediction. Finally, I just want to quickly show our investigation of into the environmental and the lifestyle factors that may have the potential to inform new strategies to potentially slow organ age. However, the further analysis needs to be done. But just here, I just show that there are something we can potentially do. For example, by personal efforts like to change lifestyle, including limited alcohol drinking and tobacco smoking, the maternal nutrition, more exercise, education, and also some efforts that would require national input, such as reduction in the socio-economic inequity, and the reduction in the pollution, and increase the residential green space and natural environment. So here, just a summary of my talk. So here, so by applying the brain age prediction framework, we performed the concurrent investigation of the biological aging across multiple organ systems. And by mapping the multi-organ aging network, we show that an organ's biological age can selectively and characteristically influence the aging of other organ systems. So we also show that we also presented the multi-organ aging profile in a range of chronic diseases. And we show that advanced biological aging extends from the organ of primary disease pathology to multiple organ systems. And in the end, we also show advanced biological age explains significant mortality risk. So just a very simple summary, we review the multi-system nature of human aging and its contribution to chronic disease and mortality. So here I would like to thank my supervisor, Dr. Andrew Zalewski and Dr. Vanessa Cropley, and also my collaborators and Michael, Breakspear, Andrea, and Nicola for their contribution to this work. I would also like to thank the UK Biobank, which this is very useful and rich data set. And thanks everyone for your attention, and I'm happy to take any questions.
Thank you very much, Yi, for the fantastic presentation. So are there any questions in the audience for Yi? Or perhaps... Christos. Maybe try, I think if you... I think it's on, but it just needs to... Oh, no, it's not on. Try that one.
great, very, very nice talk. I hate to pick on one point, but it was a fantastic talk. But I would pick on that brain age in MS population issue, which I think kind of highlights some of the weaknesses of brain age. So, I mean, obviously MS brains seem to be the oldest, but from what perspective? Typically, you know, MS patients do have some brain atrophy, but not as much as demented people, right? So, but they seem to be even older. I guess it's the presence of lesions that kind of threw off the model. So can you speak into, you know, the specificity of brain age and how do you interpret this very high brain age residuals in MS individuals?
Understand question, you mean the significant older brain age in musculoskeletal, so it implies what?
So I would expect that MS patients would not look even older than demented people.
Oh yes, yeah.
So why?
So I want to show the distribution, but I cannot go back. So from the very simplified clockwise figure, that's the brain age model trained using multimodal phenotypes. So including all the gray matter phenotypes, diffusion phenotypes, and also the functional connectivity. So I cannot remember, how about just if you only look at it, because MS is mainly affected the white matter tracks, so I can't really remember whether it appears, I think appears most, yes, it's more the white matter tracks aging. So maybe that contributes a lot to the age prediction. That's why we say that pattern. I mean, I only show just one for the overall brain age.
Thank you.
I mean, I think Christos raises a good point there. It's often hard to trace back changes in brain age, either younger or older appearing brain age, to specific phenotypes, and that might be one of the limitations of the brain age concept.
Yes, especially the UK Biobank sample is for the brain, first sample is from like 40 to 70, so that age range is not very ideal for some of the disease if we want to map that.
So we're quickly coming to the end of the symposium, so perhaps if we got any general questions for any of the speakers, there's a questioner here.
Actually, I did have a more general questions for you, but also the panel. You know, as I saw, there were, you know, 4 talks, and I think I lost count, but maybe 10 to 12 brain age models. How do you think that variability affects our ability to make inference, and what level of bias do you think that introduces in our findings, and how we interpret these different markers?
Different models, you mean the features, or the like either just linear regression or the deep learning?
Refitting multiple models, you know, one model may, you know, deep learning models versus other markers versus what markers go in, you know, free surfer features versus, you know, gray matter volumetrics, etc.
Yeah, indeed, there are so many variabilities in the like how you can train the model, the machine learning algorithm you can use. I don't think there's a very good like a direct comparison, but at least from the inference level, if we don't look at the comparison between healthy individuals and the particular disease cohort, for example, dementia. So we always say, no matter, I mean, from people from different groups, they might use their own model, but the statistics to inference generally consistent. We all see like a significant older dementia, brain age in dementia compared to the healthy individuals. So at least at the disease inference level, the overall conclusion, I don't think it will be like largely affected by which model you use. But in terms of the model accuracy, the MAE itself would maybe more like strongly affected by those selection, by the choice of models, as Christos also mentioned.
Yeah, I think it's a good point. There's certainly part of it is due to the fact that different people are trying different things. We're still learning. In the end, the community will converge. But also, there's one thing that if I were to point out one of the things that is most beneficial in machine learning models, it has been ensemble learning. So I think in the end, we will combine, and ensembles, by the way, they're inherent in deep learning by using dropouts and other things that might not be obvious, but they're hidden there. So I think in the end, the best models are going to be ensembles and combinations of multiple models.
Oh, sure. Thank you. First of all, thank you all very much for those wonderful talks, and especially the last talk. It was really interesting to. I know we all love the brains, but it's nice that you were the disruptor that talked about other body systems as well.
Thank you.
On that note, I was wondering whether that's more of a general question. Whether any of you has looked at epigenetic age. I know a lot of people are trying to use DNA methylation, for instance, to I guess assess the chronological age of humans and other animals. But I was wondering, as a more potentially accurate version of chronological age. So I was wondering if any of you had looked at it or had any plans to look at it.
So we've actually looked at epigenetic age in the Netherlands study of depression and anxiety, and indeed also found case-controlled differences in this epigenetic age gap. The effect size was quite similar, so it was 0.12 by heart. But what we're actually going to do in Enigma is to calculate polygenic risk scores for the epigenetic clocks and check the genetic correlations with the brain age gaps to see whether it kind of explains any of the variability there. We don't have, at least in Enigma MDD, we have some subsamples also available with epigenetic age, so that will be interesting to kind of see the underlying associations between those factors, yeah.
Thank you.
So we can take one more last question. Was there a questioner there at the front? Perhaps this will be the last question of this session.
Not a question. I was just answering him that, yeah, we have looked at the association between accelerated brain aging and epigenetic age in Cardia, and there is a paper published in Aging a couple of months ago with Christos. There was no association.
I think that one more last question, the absolute last question.
Yeah, thank you for your talk. It was really fantastic. I just wanted to ask, obviously we use machine learning model to calculate brain age and try to evaluate it and have a cross-validation step. I just wanted to ask if we just say ignore that, to have a good MAE, in future we have just some algorithm that's not even have a good fit. Should we have a threshold for it or something like that?
I think you should simply evaluate it on the task at hand, the clinical question, and forget about MAE. I mean, you want to have a decent MAE, otherwise the models are not informative. And I think I showed in some of my slides that if you have very underfitting models, you don't get very informative residuals, but I wouldn't worry too much about this. You want to essentially optimize your model for what the clinical question at hand rather than MAE.
So I think we've got to conclude the symposium there. I'd once again like to thank our fantastic lineup of speakers, Didac, Vidal Pinheiro, Professor Christos Stavadzikos, Laura Hahn, and Dr. Tian for presenting today. I hope the symposium has been useful. We'll be hanging around here at the front, so if you've got any other questions, please feel free to come up to the front and we'd be happy to discuss with you. So please join me in thanking the speakers again for their presentations.